Welcome students. I will continue with the chapter 5, that is acid, base and salt. We have finished acid and base in the previous video. Today I will continue with the neutral substance. This neutral substance is also called as salt. So about the acid, any substance which tastes sour is an acid, and any substance which tastes bitter is a base. There are some substances which taste neither sour or nor bitter. These are actually a neutral substance. That means the substances which are neither acidic nor basic in nature are called neutral substances. Example like distilled water. And distilled water is a neutral substance. But other than that, distilled water, take it as rainwater. This rainwater is not a neutral substance. Rainwater, it is actually slightly acidic. But of course, it does not taste sour. Those things I will tell you later on. But you remember that the distilled water is neutral, but the rainwater is not neutral, but it is slightly acidic. But sometimes even the pond water or maybe a lake water, you may find it basic. So this acidic or basic water is due to the presence of some other acidic substance or basic substance. But you know that the pure water, that is the distilled water, is a neutral. Other than that, substances like glucose, sugar, salt, cane sugar, these are some other substances which are neutral. They are neither acidic nor basic. So these are the neutral substances. So next comes the indicator. So what are these indicators, or what is indicator? Any substance which changes its color in acidic or basic medium is called indicator. Any substance which changes its color or sometimes smell also in different medium, in acidic medium or in basic medium, this substance we call it indicator. Actually, this acids are sour in taste and base are bitter in taste. But in our nature, all the substance we cannot taste it. We cannot taste all the substance, like say hydrochloric acid or the mineral acid, which are very strong. Those acids we cannot taste it. Or there are strong base also, like sodium hydroxide. These are very strong base, which we cannot taste it. Or all the substance present in our environment, we cannot taste it. So we have to use some way or technique to know that medium, whether it is acidic or basic. There we use indicator. So indicator are those substances which changes its color in acidic or in basic medium are called indicators. The same substance, that is that particular indicator, will give different colors in different medium, that is acidic and basic medium. They shows different color. Sometimes there is another type of indicator which changes its smell in acidic or in basic medium. Simple example like clove, or it may be onion also. This onion will give different smell in acidic medium or in basic medium. So by knowing that particular smell, we can identify that the medium is acidic or basic. So some of the common substances, the substance which we find it easily and use it as an indicator are like say turmeric or litmus, China rose petal, red cabbage juice. These are some of the naturally occurring substance used as an indicator. So as a whole, we call it natural indicator. The next one is artificial indicator. Artificial indicator means those indicators which are manufactured in the industry, and these are specially manufactured as an indicator or to know the acidic or basic medium. Example is phenolphthalein and methyl orange. These are some artificial indicator. Come to some natural indicator like say litmus. Litmus is an indicator. Actually, this litmus, it is extracted from the lichen. This lichen generally it is grown on the bark of a tree. So from this lichen, litmus is extracted, and this litmus we use it as an indicator. Actually, after extractions of this litmus from the lichen, we make a solution, and this solution changes its color. Like say in distilled water, it will give purple in color. So if it gives purple in color, then it is a neutral medium. Now, how does this litmus solution work actually? When this neutral litmus solution is added to an acidic solution, it turns red, and when added to some basic solution, it turns blue. So this change in color, you should remember that when it changes into red, that one is acidic medium, and the same litmus solution, when it changes into blue in color, then it is a basic medium. Suppose say you take two medium in glass 1 and in glass 2. These are different medium. We want to know the contents in that glass 1 and glass 2 are acidic or basic. Then we use this litmus solution. If we put this litmus solution in that glass 1, and if it converts into red in color, that means that medium is a acidic. In that glass 2, if you put the same litmus solution, and if it turns blue, that means the medium or the substance or maybe the solution that is in that glass B2, that is a basic medium. So in this way, we can identify different medium, whether it is acidic or basic, with the help of litmus solution. This litmus in the laboratory, it is present as a red litmus solution and a blue litmus solution. In the laboratory, whatever litmus it is present in the form of a red litmus solution and blue litmus solution. This red litmus solution, if we put in a medium and if it converts to blue, then that is basic. That means red litmus is converted into blue in basic medium, and blue litmus solution is converted into red in acidic solution. So in this way we can identify. The other form of litmus that we get in the laboratory, it is as a thin strip of paper. There are two types. One is blue litmus paper and the other one is red litmus paper. The color change will be same. Like a blue litmus paper will change into red. It is blue to red. It is acidic medium, and when it changes from red to blue, it is basic medium. So with the help of that natural litmus solution, or maybe that litmus solution, red litmus solution and blue litmus solution that we find in the laboratory, with the help of that also, at the same time the red litmus paper or the blue litmus paper that also find in the laboratory, with the help of this litmus, maybe the litmus natural solution or the litmus solution of red and blue or the litmus paper, with the help of this we can identify a medium whether it is basic or acidic. In your book, in the table, some examples are given, like say lemon juice. If it is lemon juice, effect of red litmus paper, in that lemon juice, if you put in this red litmus paper, or if you put a drop of the lemon juice in that red litmus paper, then there will be no change in color. But if you put a drop of lemon juice in a blue litmus paper, then it will change into red in color. The blue litmus paper will change into red in color, because we know that lemon juice is acidic. So in acidic medium, blue litmus paper changes into red. So it infers that it is an acidic solution. Or suppose a common salt solution. Common salt means sodium chloride that we find at home. So put little amount of common salt in a water, in a pure water, and dissolve it. That becomes a common salt solution. So either you put the red litmus paper in that solution, or take a little amount of that common salt solution and put it in that red litmus paper, there will be no change in color. Or when that blue litmus paper is dipped in that common salt solution, there will be no change in color, because sodium chloride is a salt, it is a neutral medium. That is why there will be no change in color for both the blue as well as red. So these are some of the examples that is given. Second one comes to turmeric as an indicator. This turmeric, it is also used as an indicator. Since turmeric we find it naturally, and so it is called natural indicator. How we can use this turmeric as an indicator? What we can do is, we can make this turmeric into a turmeric paper, just like that litmus we can make in the litmus paper. So here also this turmeric, we can make a turmeric paper. To prepare this one, what we do is, we take little amount of turmeric powder and add again little amount of alcohol in this turmeric and make a paste. This paste is spread on a blotting paper or a filter paper, and when it dries, that becomes the indicator paper, that is turmeric indicator paper. The yellow paper, it will be yellow because turmeric is a yellow. That one we can cut into a thin strip, and that one we can use as an indicator. Now in case of turmeric, basis turns yellow turmeric into red in color, but acid and neutral medium, there will be no change in color. There will be no change in color for neutral medium and acidic medium. Suppose say there is a particular solution, there is a particular solution or a medium, and we want to know whether that medium or a solution is acidic or basic. Now if we take that paper containing turmeric or made from the turmeric, if we dip in that solution, and if that yellow turns into red, then we know that that solution is basic. But if there is no change in color, then that solution, it may be acidic or it may be neutral. So This yellow turmeric pepper, it is mainly used for indicating a solution whether it is basic or acidic, or most appropriately for identifying basic solution it is used. So here in the table again some examples are given: change in color of the turmeric pepper and nature of the given solution. So first example is given about soap solution. So this soap solution, in that soap solution, if you put in that yellow pepper, it will change into red. That means it indicates that soap solution is basic in nature. But if we take shampoo solution, if we take a shampoo solution, the shampoo that we use, shampoo solution, and if we put this turmeric pepper, then there is no change in color. That means that shampoo solution is either acidic or neutral solution. Or the other one, like say dilute nitric acid. Nitric acid is a mineral acid that we know. So when we dip a turmeric pepper in this dilute nitric acid, there will be no change in color. That means it is acidic or neutral solution. But nitric acid, we know that it is a acid, a very strong mineral acid. So come the third one. The third type of indicator, it is China rose indicator, and more are there, like say the artificial indicator like phenolphthalein and methyl orange. About this indicator we'll discuss it in the next class.